Okay, class. So I am your instructor. My name is Louis Obermiller. I received my PhD in molecular and cellular biology and biochemistry from Arizona State University in 2000. So I generally am an expert on things that are small. And so I teach classes that deal with genetics and microbiology and enterobiology for majors and things like that. So this is the intro to the class for fall of 2020. This is kind of a unique class because it is going to be completely online. Bio 181 really wasn't meant to be completely online because there's some lab materials that are much better for you to learn hands-on. It's kind of like trying to teach someone how to play guitar in the air. But because of the current situation we're in, it is how it is, and so we're going to do the best that we can. It's been kind of a whirlwind summer. Faculty aren't contracted to teach over the summer, so we found out that everything was going to be online, and generally the campus is going to be closed for the fall. And so when you go through these labs, if you notice any mistakes or anything like that, let me know. Okay, so we're going to start from the beginning. And so that means we're going to go to www.mesacc.edu. And when you do that, you should get this screen. And on the top, and you can probably see a yellow circle. I can't see that on my end, but that's just showing you where my mouse is. And when I click on something, it becomes blue. So the way that this course is going to be delivered is through Canvas, and so you want to click on this Canvas tab, and it will bring you to Canvas. So you will have an MEID that you've been given through the district, and then a password for that, and you just go ahead and log into Canvas. And then when you log in, you'll see your classes in here. I'm teaching 3 lectures, and each of these lectures have 2 labs. So anyway, they're all exactly the same. I'm just going to click on this one, but it doesn't matter what section you're in, to go ahead and open it up. So this is an older video. I posted this for people because they were asking about text and things like that. And this video that I'm making now will replace this old video. It'll take a little while for Google to caption this. So just bear in mind that if it doesn't have captioning, Google usually takes about 24 hours from YouTube to put it on there. So I'm just going to go through how the course is laid out so that you can sort of see this. And I've published this course. This view is going to be different than your view. You will have a student view, but generally it's going to be very similar to my view. So anyway, there is a virtual tutoring center, and you can click on that link. It will open it up in a new window, and you can get tutoring for biology. Individuals may or may not be online, so you can see if they're offline or online. And then if you have any questions, any of these classes, any of these tutors should be able to help you if they're available. And again, I'll have office hours as well. And occasionally I'll be on here helping tutor also. So that is pretty much it. I'm going to go back to home. This is the link to my office hours. I don't like WebEx, so I'm going to use Zoom. I have a Zoom license, so I'm not limited to the 40 minutes that most people are. And you can click on this and then click on to join my Zoom meeting. You'll need to download Zoom. It'll prompt you to do all this stuff. So if you want to come to office hours, you can do that. It's pretty much an open meeting. It won't be recorded. and I can put people in a waiting room in case there's something private that you want to discuss during the Zoom meeting. So anyone can join any of the sections. That's when I'm going to be available to answer any questions that anyone may have. All right, so let's go back home. Again, these office hours are not mandatory. It's only if you want to join. Some students may not be familiar with Canvas, so one of the things, one of your assignments that you're going to have is to post a profile picture in Canvas. So if you wanted to look up how to do something in Canvas, you can just look it up by searching through the Canvas guide. So this is how can you add as a profile picture as an instructor, and this is how you can add a profile picture as a student. And it'll tell you take a picture, you can edit that picture, and then upload that picture, and then resize it. And then, of course, I would like the picture of yourself so that I can put a name to a face. So don't put a panda on there, uh, or a picture of your cat or whatever. I'd like a picture of you where I can actually see uh, what you look like, so I can sort of know who is in this course. It's kind of helpful to sort of, under the circumstances that we're in, that we won't actually be seeing each other. And you'll see plenty of me through lecture videos. I'm sure that you'll be sick of that by the end of the semester. Okay, so anyway, any questions that you have, you can go through the student Canvas guide. This is how the lecture videos will be posted. So these lectures are from live classes that I taught. You'll see some stuff written on the whiteboard, but don't worry about that. If you can't see it, I know it's hard to see, but everything that I'm writing on the whiteboard is the same stuff that's generally on the slides. And I'm going to try to update these videos so that through Camtasia, so that I'll have um, a virtual whiteboard that you can actually see the stuff that I'm writing on. But it's really nothing different that's on the PowerPoint slides. But if you have any questions or anything like that, just jot down some notes and then visit me in office hours, or you can shoot me an email. All right. So Panopto, this is a software that's purchased by the district, by MCC, and each class should have these videos. So there's two sets of videos from two different classes, and you can pick whichever ones you want. One thing about Panopto is it only shows 25 videos at a time. So if you click on 50, it'll show you the other set. So this set is from dates, and this is from 2016. These are slightly newer videos, but essentially it's the same material. It hasn't really changed at all. So we'll still be covering the exact same material that I covered last year. So let's say that you wanted to start here at the beginning, chapter 1. You just click on that, and then it'll bring you to a screen, and there may be some things here. Try to pause this so that so anyway, this is the whiteboard and this is me, and I know that you won't be able to see that, but don't worry too much about it. This is the screen capture, so this is everything that's on the screen, and then these are the PowerPoints slides that I go over. So if we just went on to this class a little bit, we can fast forward it. Clone Michael Jordan. They believe that we were visited by aliens millions of years ago, and they gave us the technology to clone ourselves so that we could raise clones and take the information out of our brains and put them into our clones forever and ever and ever. So that's the company Clonaid. They actually claim to clone the first human being, and we'll talk a little bit about cloning during this class and how that's accomplished and things like that. So anyway, if you just continue through this, you could fast forward through it if you want to different slides. And again, you can see that I'm writing stuff on the whiteboard, but it's exactly the same as what's on the slides. So anyway, um, so you can go over the lectures in Panopto. One drawback about Panopto is that it's not very good at captioning. So it tries to caption, but it's really not nearly as good as Google. You probably know that. So anyway, if you need captions for whatever reason, you'd probably be better off not using Panopto. I kind of like Panopto because it has these three different split screens. But the YouTube videos are all these combined together. So basically, you see this, uh, my screen, and then there'll be a small insert of me, talking. But you don't really need to see me for this class anyway. So I'm going to close this out, and then these go by each of the lectures when they begin for each of the chapters, as you can see, and then this ends. So this is the last one, final exam, and then these are another set of videos, but they're only done by date. They're not done by the lecture. So most students prefer to go by the ones that are labeled by the lectures in each of the chapters, and that's what I recommend. Okay, back home. Here's the link for the YouTube videos. These videos are mixed together with some genetics videos, but I did make a playlist, and so for 181, and so you can go through that. Here's the playlist, and it goes through the intro. This is from 2019, from spring 2019, and this is that same video that you'll see the link to before I put this new one up. This is an older intro, 2018, I think. And then again, it just goes through each of the lectures all the way through to the final exam. There's genetics practice problems when we get to chapter 14, and these are the solutions for those problems. So you can click on that, and I solve the problems on what you've learned. Hopefully, you've already watched the chapter 14 video and the chapter 13 and the 12 lecture. But what we know is that. So make sure that you guys watch the chapter 14 lecture here before you attempt those. Except up. So for glucose to come in, and then glucose comes into your cell. Right? And that is the first step from this glucose, that hexokinase. And they're not ordered here, but they are. Those are the YouTube videos, and you can watch those as well. So I'm going to go back home. Again, it's meant to sort of simulate the exact same thing that you would have if you were going to an in-person class. So, there's students there. They're going to ask questions. The audio isn't really that great. The cameras sometimes fail, but it is what it is. And I'll make a new set of lecture videos that just have a better quality audio and video to them as the semester progresses. All right, so each chapter has PowerPoints, and you can click on those PowerPoints and you can download them. So every student has an account to Microsoft Office Suite 365. If you registered at MCC, then you can call the Help Center and they can get you hooked up with that. Canvas attempts to decode these in Canvas itself, and so you can go through the slides here as well if you want to go and review them, and you can also download them. So that's available to you for each of the chapters, chapter 1 all the way through chapter 17. We won't cover everything in the textbook. It's just not enough time in a semester to cover every individual chapter, but I will cover the things that I feel that are most important for your future classes. Most of you probably going in the medical profession, so that's sort of what focus on. This class is somewhat difficult because you're learning biology and chemistry, and you're also learning a foreign language at the same time. And that foreign language of science is Latin and Greek, and sometimes it's German. Um, but there are some terms that you need to know because you have to learn how to speak like a biologist. It's kind of like if you were in a hospital and they said, bring me some Pitocin stat. If you didn't know what stat meant, you might just be standing around doing nothing. Stat actually comes from statum, which is Latin for right away. So um, that's sort of how biologists speak. I thought that it would be more fun to do crossword puzzles than to just make flashcards. But if you want to make flashcards, you can do that. These are not turned in for a grade; they're just study guides for you guys to learn the terms. So, two across the complete genetic code of an organism is actually genome, and then I have the answers to each of these crossword puzzles here, so we can check our work and see if two across is, sorry, that's the crossword. We need to go down to the answers. And we can check our work, and two across is indeed genome. So you can check your answers there. Every chapter has a study guide. So you should, this is, again, I don't collect this and I don't have the answers. I want you guys to try to figure this out yourself. So I don't want to just sort of spoon feed you the answers. But if you have questions, like if we're going through chapter 2 review questions, substance cannot be broken down to other substances by chemical reactions. Carbon, hydrogen, nitrogen, oxygen. So these are the four most common elements in living systems. It talks about that you should know a neutron is no charge. Its molecular mass is 1.009 daltons. And all this stuff is covered in the PowerPoint or the lecture. So one thing that I want to caution you on is just studying the PowerPoints, because the PowerPoints are just meant to be a guide for me to talk about things in greater detail. So I couldn't put everything I wanted to say on a PowerPoint because it would look just like me showing you a picture of a textbook page. So it's just an outline. So make sure that you guys, when you're studying the PowerPoints, you also listen to the lectures, because the exams come from what I say, and that's based on what's on the PowerPoints. So make sure that you listen to the lectures, and don't just try to study off the PowerPoints, because you won't do well. Okay, so we covered the videos and the PowerPoints, the crossword puzzles, the answers for those, and the study guides for every chapter. Those are the genetics problems that we were talking about, and there are videos for them. So after we get to chapter 14, this will make more sense, but there are genetics problems like, black guinea pigs from the same genotype. There's a fancy Greek word, gene, just the type of genes that it has. Were made to produce 29 black and 9 white offspring. What would you predict the genotypes of the parents to be? So there's a difference between genotype and phenotype. Phenotype is what you see. Genotype is what your genes actually are. And those two can be different. So anyway, by the time you're done with chapter 14, you'll be able to solve these problems relatively easily if you pay attention to the lectures. And I'm going to go back home and I'll show you the answers to these questions are here. And I've written them out. And again, I made videos for them as well. And that's on that YouTube channel. So this would show you that their genotype, they would have to be big B, which represents a chromosome that would produce a black pigment, and a little b, which represents a chromosome that is broken and produces no pigment, and both of them would have the same genotype. Their phenotype would be black, because you only need one dose of black to have black fur. And again, you'll know that once we cover chapter 14. Okay, so let's see. These are audio recordings from the fall of 2017. I haven't really just recorded audio, so it's generally been video since then. But some students have asked me for audio so that they could, download it to their phone as an MP3, or they could listen to it while they go to the gym or walk around or whatever. So I put these on here, and you can listen to the lectures as well. The lectures are the same as the video lectures, even though they're from 2017. There hasn't been a lot of new things. I mean, there has, but it's all based on old concepts. So that's the homepage. Uh, for announcements, I'll have announcements here, and one of the first announcements I'm going to make is that I've made this video, and I want you guys to watch this so that you understand the contents of the course. I might also make an announcement like my kids are sick, so I can't make office hours or things like that. So make sure that in the email it gets sent out when I make an announcement. So make sure that you check your Maricopa email, and also check the announcements if something weird is going on, like I don't show up for office hours. Okay, so here's your assignments. This is everything that's due. And I'll show this in the syllabus as well, but there are quizzes for every chapter. Um, there are 15 chapters that we're going to cover, so we're going to skip some. There are some things that are hidden here. You guys will see all the things that I've checked in green on your end. So the quizzes are worth 10 points each. The rule is that I drop the lowest 5 scores from those quizzes. So I only count your highest 10 grades off of these quizzes. Now, if you click on a quiz, it'll open it up in a new window, and you can, it'll tell you to start the quiz. I'm just previewing it because I'm the instructor. But you can go through this. Which of these categories of organisms is the least broadly inclusive? So the least broadly, the most inclusive. What are archaea? Thanks, Norton. What if you think of something that's archaic? It's from the Latin, which is old, and so these are organisms that lived on the Earth when it was really a rough place to be. It was super hot, lots of salty environments. So Archaea Bacteria are the first living organisms that we know of that lived in these really extreme environments. So make sure that you read the questions carefully, right? They didn't only live in salt environments. They lived in really high temperature environments like volcanic springs, Yellowstone, and stuff like that. So you couldn't pick this one or this one because that it's a broader group than that. So the best answer here would be prokaryotes that are extremophiles, which means they live in really extreme environments, and they share some bacterial. When we say bacterial, we mean eubacteria. Eu means true. This means true as well. Karyote is nucleus. So a eukaryote would be an organism that has a true nucleus. Bacteria don't have a nucleus, and you'll learn that whenever we go through these chapters as well. So when you take the quiz, you go back to assignments. So when you take these quizzes, you get 3 attempts. You have 20 minutes for each quiz. And then again, I take the highest grade. So this is kind of a give me. There should be no excuse for you not to get 100 points because I'm dropping the lowest 5 grades, and you get 3 shots at each one of these. And it's completely open book. So, but the reason I give you these quizzes is because the questions that you see on these quizzes are very similar to the questions that you'll see on the exam. So the exams are multiple choice, just like this. There's going to be 5 answers, just like this. So, um, I don't want students to just come in and start the test and go, I have no idea what it was going to look like. This is meant to show you what the questions on the exam will be like. And in fact, some of them will be the same. So it's in your best interest, even though you get 10 points, let's say the highest grade on one of the quizzes, there's a question bank that this gets pulled from. And so you might see different questions every time you retake the quiz. So if I were you and I wanted a really good grade, I would take all 3 of the quizzes, even if I got 10s on all of them, because you're going to see questions that you might see again on the exam that are going to be really similar. And that's a hint since I make the exams. All right, so let's go back to the assignments. And there's all the quizzes they're due the day before the exam. I don't want you guys to wait till the very last minute, so I'm going to make them do one day before the exam. So the first exam covers chapters 1 through 5. Your quizzes are due on the 29th at 1 minute to midnight, and your exam is on the 30th, also due at 1 minute to midnight. These don't require a lockdown browser, but your exam will. All right, this is just for me. All right, these are the at-home labs. So 100 points, the whole class is worth 1,000 points. 100 of those points come from quizzes, and then 300 of those points come from these labs. So normally you would come in and do some of these labs that require expensive equipment, like multi-thousand-dollar microscopes or spectrophotometers or centrifuges or what have you. But because of COVID, they're not allowing students on campus because it's too hard to disinfect that equipment and it's super expensive to do that. And so we're just going to do the best that we can using some online resources to try to sub for that. So the first thing that you're going to do is a chemistry lab. So you'll click on that, and then there'll be a Word document link. So you don't need a lab manual for this semester. Everything's online. You click on that. It'll download it, and you can open it. And then just read through the lab. So the lab will talk about functional groups. Here's some functional groups here. A hydroxyl functional group is one that has an O and an H on the end of it. You'll need to know that. That's super important. Carboxyl groups are groups that are C double bond O to an O and an H. So this isn't a hydroxyl because it has this specific group on it. Amino groups. This is where amino acids that make proteins get their name. So they have one of the ends is this nitrogen with 2 hydrogens on it. And that's an amino. Phosphate groups are super important in DNA. This gives DNA its negative charge. And you guys will see that forensic scientists and 23andMe and all these other companies that look at genetics use this negative charge on DNA to pass the DNA through some sort of substrate that can separate it out based on its charge and its size. And that's how they can figure out your DNA sequence and if you have a mutation in, say, the breast cancer gene or the prostate cancer gene or something like that. Or even if you have COVID. Okay, so this is a carbonyl group. Carbonyl group is a C double bond to an oxygen. There are 2 kinds. There can be one in the middle, or this one's on the end. So this is an aldehyde, and then this one is in the middle. So that this R just means that there's more carbons linked to it. This one means there's carbons, but this terminates. You'll learn this in chapter 2 that hydrogen can only make 1 bond. So it's kind of like having putting a stick in an orange and not having any other holes to put other sticks on. So that ends your ability to build anything on it. And that's why this is a terminal, because a hydrogen terminates. It can only make one bond. Again, here's another one. So this ends it, and this R just means there's lots of other stuff there. So this is a ketone, this is an aldehyde, and this is really important in sugars and how we process sugars. And you'll learn that later on in the semester. The purpose of this lab is to get you used to a reaction that builds molecules and breaks them down, because that's essentially what we're doing. So, when you eat food, you're breaking down the proteins, you're breaking down the sugars, the carbohydrates, you're breaking down the fats, and you're releasing energy from that. And so that's how you're able to live. And so, because we're studying life, that's kind of important reactions. And then, you know that you can convert carbs into amino acids, protein that makes muscle. So those reactions require you to remove water to build molecules. We call that anabolic, like anabolic steroids. Ana means up, and so like Hans and Franz pump you up. And then if you want to break something down, you have to add water into it. So we call that a hydrolysis reaction, or if you pull water out, it's a dehydration reaction. Anyway, so that's the purpose of this lab, is to teach you about that. And so you're going to circle the functional groups, and then you're going to do the reactions. You can see water comes out if you're going to build protein, right, from two amino acids. Again, this is a carboxyl group, and then this is an amino group, and that's where it got its name, amino carboxylic acid, so amino acids. Two amino acids, glycine and alanine, make what we call a dipeptide, a protein with two amino acids, and to do that, we have to remove water. And that's kind of interesting, and I'll talk about this in lecture, but a lot of people believe that life formed in the ocean, but it's really hard to scientifically think about that, because when you put DNA in water, it tends to break down because water breaks molecules down. Like if I was a forensic scientist and I was at a crime scene and I was collecting evidence, I wouldn't want to put that DNA sample in water because it would break down. And scientists don't do that. So it's hard to imagine how life could form in a water environment. Anyway, so once you guys fill out the answers to each of these, it'll ask you what's the functional group on glucose and so on. So you can scan this. There's a CAM scanner that a lot of students are familiar with. You can scan it with a CAM scanner into a PDF. You can take pictures of it as a JPEG, however you want to do that. And then once you're done, once you fill that all out, you're going to submit it on Canvas. So I'll show you how to do that. I don't know what it is. So I can't submit it here, so I'll go to settings and show you the student view. So we'll go back to the chemistry. And you'll get this blue box that says submit assignment. So once you've done that and you've taken pictures of it or scanned it or however you want to submit it, remember, if I can't read it, I can't grade it. So make sure that you guys double check that your pictures aren't blurry, your scan isn't blurry, and all the pages are present before you submit it. And then you submit it just by attaching a file. So you just like if you were attaching a file in an email. One thing to keep in mind is that once you submit this, it goes into a file. But if you resubmit it with the same file names, it will erase the files that are already in there. So make sure that you guys, if you are resubmitting it, chances are it's going to delete everything else. So if, let's say, you forgot to include a picture, you have to resubmit the whole lab again. You can't just submit one picture because it'll erase everything else that you've already done. A lot of students just take their pictures and drop it into a Word document, and just make sure that it's big enough that I can actually read the answers in it, because uh, I don't have a microscope that I can put up to my screen to read. So, anyway. And then you just submit the assignment. All right, so it's pretty straightforward. I'm gonna go back to assignments, and so you have 14 labs that you'll do exactly the same way. Every one of these labs has a link, right, that you'll go to, and you'll open it, and then you'll complete the lab, scan it, the whole lab, right? You can use a cam scanner or take pictures of it, or your answers in there, or if you want, you can do it in Word, and then save it, and then just submit it. Okay. So each of these labs are worth 25 points, which it shows here. The rule is that I drop your two lowest lab scores. So let's say that you forgot to do a lab, or it's just a super crazy week or whatever. Um, you are allowed to skip two of these labs without penalty. After that, you're going to lose 25 points. And then 12 times 25 is 300. So that 300 points of your score will come from the labs. Some of these labs require advanced prep, like plant variation lab. You're going to plant some plants, they're going to grow, so that's going to take a couple weeks. Then you're going to take the seeds out of those plants and you're going to plant them again. It's a genetics experiment, and then those are going to take a couple weeks to grow. So the whole lab itself takes about 6 weeks. So you're going to need to start that lab on September 14th to get it done and to get all your data to submit it by December 14th. So that's when that's due. And I imagine a lot of people will decide to skip that lab, which is kind of a shame because it's fun. It's a fun lab, and it will teach you something about genetics, which is kind of a complex subject. But the bacteria lab, that takes a week. You have to pour your agar plates first, and then you're going to swab stuff like keyboards and money. And you can see how gross some things are in your house. And then the bacteria take about a week to grow on the plate. So you're going to have to start a week in advance so that you can get your data and then submit the lab. So you'll want to start this lab on September 14th, even though it's due on the 21st, because it takes a while to prep that. The Osmosis Lab, you guys are going to deshell some eggs using vinegar. And so that takes about 2 days to sort of the vinegar to eat away. It's a carbonic acid reaction. So vinegar is acetic acid, and it's gonna turn that calcium carbonate, uh, and it'll bubble away, and then the eggs will be removed, and then you can do the osmosis lab. But you're going to need to soak those eggs about 3 days before you can actually do the lab. So even though it's due on October 5th, you want to make sure that you start that around October 1st, and then make a couple extra eggs in case you drop one. Other than that, some of these are going to require you to watch some videos. So like the Sonoran Desert Plant Lab, you'll have to watch videos that are done by some colleagues at MCC. So you'll have to follow these links. It hasn't been hyperlinked in there, so just copy and paste it. All right. There's some extra credit. So like I said, if you post a profile picture on Canvas, I'll give you 10 extra credit points. It doesn't tell you the point value here, but I can tell you right now that this is what each of these, this one is worth 10, 10. I'll add that in. And then this is 10. And then this one is 25. You don't have to actually donate blood. You just have to make an attempt to donate blood. If they refuse you for whatever reason, then just get a note, um, and I'll give you credit for that. Your exams. So the exams haven't been made, but you'll see this whenever they get created. The exams require you to do a lockdown browser, Respondus, which is right here. So I'm going to make this first quiz a lockdown quiz so that you guys can practice using the lockdown browser. It's going to require you to install some software, and then it will record you as you take your exam. So make sure that you're in a quiet place. You don't have any other electronic devices around you, and that it's meant to simulate an in-person exam. So each of these exams are going to be worth 150 points. And like I said, they're going to require Respondus. I'll give you guys a chance to practice on Respondus with one of these quizzes so that you can see exactly how that works and to make sure that you have all of the equipment that you need. The most important thing that you need for Respondus is the browser itself, and then you're going to need a webcam and a microphone. So if you don't have that on your computer, you should get one, because you can't take the class without it. And that was in the notes whenever you registered for the class. All right. So there's discussions. You guys can start discussions in the class. I've already started a discussion about population genetics. And so you can see that there's already been one post. And so if we look at that post, you'll see it's asking you to help solve the genetic mystery in humans. So there's Mendelian traits that include free earlobes, which means that the earlobe is very distinct, or attached where it really is not. And then you have a bent little finger or a straight little finger. And so I want everybody to answer this because you're going to need to do that for the lab, for the population genetics lab. We're going to need data because we need to do it on a population. So here I've responded, I have a bent little finger and I have an attached earlobe. And when we get enough people to respond to this, then you'll do that, and I'll just show you. We go back to assignments, and then we go to the population genetics lab. Download that. This is the background on it. Though some of the labs require you to go to the store and buy some stuff like Reese's Pieces and things like that. So here it's asking you some questions, and so those questions are: in humans, free earlobes is caused by dominant. So count the number of people in your lab section with free versus attached earlobes, and you're going to write it in, and then you're going to do some math. And from that math, you'll be able to calculate what the frequencies of each of the alleles. So you remember we have two genes. The technical term for those are alleles for every trait. So you like if for say brown eyes, so you would have one allele you got from your mom and one you got from your dad. And let's say your dad gave you brown, your mom gave you blue. Well, brown's dominant to blue, so you would have brown eyes. So you can calculate that based on populations. And then there's an example here that we can use for real world things like if we go to hospitals and we find out how many individuals have a certain phenotype, a disease, you can calculate the risk that any one individual in a population would have that particular allele. In this example is cystic fibrosis, but it works for anything. And it's kind of a powerful technique. So this explains how you can calculate that. So make sure you guys read over that, and then use the data from that discussion to enter in those values. All right. So anyway, if you guys want to make a new discussion board, you're like, I don't understand this topic or whatever, you can discuss these things among yourself. Sometimes I'll jump in there. If you guys are having a problem with something, I can go in and answer it. You can get your grades. I'm not going to click on that because it'll show everybody's names and information. Um, and I think that you have a link for the individuals in the class, but I'm not sure. Pages, these are exterior links, web links that will help you out. So one of the things that is kind of nice is this biology crash course because it's really good for visual learners because this has high production value. They make millions of dollars off of YouTube views for this, and they have green screens and a whole crew, and so they can do things that I can't, and the school wouldn't provide me uh, with the financial means to do this. But it can go through.
If you're wondering, this is how the most revolutionary course in biology of all time begins. Come today to learn about covalent and ionic and hydrogen bonds. What about electron orbitals and the octet rule? And what does it all have to do with a madman named Gilbert Lewis? It's all contained within.
And then there's other links. So this is the Qriket assignment. You have to enable Flash Viewer to do it. I want you to do the tutorial and the Qriket, and then it'll go through each of these. Um, and it's generally a scientific method. So I'll give you a freebie. Uh, when we talk about scientific method, how many different methods are there? And the answer is many. It's like asking what's how many different ways are there to get to Flagstaff, or how many different ways are there to get to Amarillo, Texas. Um, you can go about the scientific method in different ways. What is important is what the end result is. So that's the correct answer. All right. Properties of water, different things. 23andMe's, people are interested in. Some of these links are broken, and I'll go back and I'll try to fix these. Um, but DNA transcription, RNA splicing, how things move in and out of the cell, and then different PowerPoint viewers and things like that. You don't really need this anymore because they now provide you with Office 365, so you can get that there. And if you go back to the pages, there are links for videos for some of the labs, like the metric system lab that you'll do, how to pour petri dishes. So this will explain to you exactly how.
Growing bacteria is a very popular science project, and it's really simple to do. This video will show you how in just 3 easy steps, plus show you some pretty cool bacteria cultures.
So you guys can look through that and get some tips and tricks about how to do your at-home labs. All right. I'm going to skip over the syllabus for right now. I'll come back to that. Study mate, that is a flashcard making thing, so you can do that if you want, or you can make flashcards the old way, but there's a video on how that works. This is a link to the same quizzes that are under assignment, so if you click on that, it's going to take you back to the exact same list of quizzes. Some of them are hidden because they're exams, but you'll see the ones that have the green check next to it. There's chat, so you guys can chat with each other if you want. I generally don't go on that because I'm doing Zoom office hours, but if you are online and someone else is online and you're like, I don't understand this electron orbital thing. What's the difference between valence and valence electrons? Then you can ask. This is the link to the lockdown browser. Your thing is going to be different than mine, but it shows you how to do it. Here's another link for online tutoring. So there's a thing called BrainFuse that you can use. It's going to give me a different dashboard than you, but like you can get live tutoring. There's different classes, and some of the tutors are unavailable. They're actually pretty good. I've tested this out, so I would be pretty confident in any answers that they would give you guys. Okay, so that pretty much covers everything. I'm going to go over the syllabus real fast so that you can kind of see what's going on here. Um, and then again, just remember you have 4 exams. Those 4 exams are worth 150 points each, so that's 600. So 60% of your grade is from exams, 30% or 300 points is from labs, and 10% or 100 points are from the online quizzes, and that's your grade. And then you can do extra credit if you want to try to bump up your grade. Okay, so this is the syllabus for 181. These are the lecture sections. So there's, like I said, 3 lectures that I'm teaching for fall 2020. You should be enrolled in a lab section. So this lecture section has these 2 labs. This lecture section has these 2 labs, and this lecture section has this 1 lab. So make sure that you're enrolled in the lab and the lecture. You have to take both because you can't get credit for one without the other. I'm your instructor. That's my name, Louis Miller. I have a PhD, like I told you, in molecular biology, molecular and cellular biology, and biochemistry. Um, this is my office phone. If you call and leave a message, it sends me an email with that. So it's a lot better than it used to be. I have to go through some really crazy procedures to go on campus. I have to call public safety, and then I have to wear a mask and from my car all the way in, and then they have to go and disinfect everywhere that I've been. And so I probably won't be on campus very often. So we're gonna do most of this stuff online. My office will be online, like I said, 1 to 150 and via Zoom, and that link is in Canvas. And then Friday by appointment. So just let me know if you can't make these office hours, and I'll try to work something out with you. Lectures online, like we said, and you've already seen the Canvas things. Make sure that you do the. So we talked about learning this material. The material comes from what I say, and that's based on the outline from the PowerPoints. The crossword puzzles cover the terminology that you need to know. So the words, the like foreign language component of biology. The study guides go over the things that you need to know that I covered in the lecture, and then you can go over those things either using MP3s, those audio recordings, or video recordings, the MP4s. And then, like I said, those are available on YouTube and also Panopto. The exams will also be online, and you will be required to use the lockdown browser. So you need a webcam and a microphone for your computer. There's no exception. So if you don't have one, you're going to need to go buy one. Um, Labs are all online, and we'll follow the schedule in Canvas. And there's a lab due every week, except Thanksgiving week. So that's the 1 week that you get a break from the lab. You need to order a lab kit from the MCC bookstore. I think it's $18.75. I just put approximately $19 because I don't know if that's going to change. Originally, they were going to allow you to come and pick them up at a certain time in the parking lot, but they're running into the same issue as I am, where they, have to decontaminate people and things like that. So they're not going to do this pass them out in the parking lot. They're going to require everyone to have their kit shipped to them since the campus is closed. So, like I said, there's no pickup option. You need to order this lab kit by September 1st so you can have it by the 7th. That's when your first labs are going to start. And if you want, you can click control and click this link. And that's very small. Oh, so anyway, all the kits are the same. So every single 181 class, no matter if you're in my section or a different professor's section, which you probably won't be watching this video if you were, but they're all the same. So you just click on, there's no used option, add it, then you'll go to checkout. There's different ways that you can pay for it. I don't know if there's a promo code or not. Anyway, and then I think that you can go to checkout. Um, there's no replacement. I think this is just a default thing. So just whatever it defaults to. You can check out at a guest or whatever, and then it'll ask you to deliver it. And I think I'm not going to go through this whole thing, but I think it's $7.50 to ship it. So you're looking at somewhere around 25 bucks, 26 bucks for the lab kit. And that's going to come with all the stuff you need to grow the bacteria and the scales for the metric system and the beakers to do the Osmosis Lab for the deshelled eggs and all that other stuff. We thought it'd be easier than everyone running around all over town trying to collect all this stuff like clay and every these other things that are hard to find, like beakers and graduated cylinders and things like that. And so, it's it's a lot cheaper than what it would cost you to go out and get individually. So, because they bought it in bulk for like I think there's over 1,181 students, so they got a pretty good deal on it. Anyway, so that is the lab kit, and everyone needs to order that. The books, so Campbell Biology is the book that we use, but there's a lot of other options now, like OpenStax. They used to produce a textbook that was a half textbook because we only cover the first half of the book in 181, and then the second half of the book is covered in 182. But I don't think Campbell makes that custom edition anymore. So they're on the 12th edition. It just was released. Um, they don't change a lot between editions. I've been teaching this class since the 6th edition, so I'm sort of dating myself. But um, the if you get anything above the 9th edition, then it's basically the same as the 12th edition. The only thing that's different are the pictures. And normally I tell my students to go to the library and look at the books because I have them on reserve. But you guys can't go. I don't think you can go to the library, so you can't. You're just gonna have to take my word for it. 9th edition is fine. I think it's 20 bucks, maybe $5 shipping on Amazon. So you can own the book for 25 bucks, or you can rent it for the same amount of money. It's up to you. A lot of students say, "Do I really need a book?" The answer I give is, well, no. But most students do about a letter grade lower when they don't have a book and they're trying to use Google and stuff like that. So, is it worth the 25 bucks to get a book? Yeah, I wouldn't pay 150 for it, but I think 25 is a reasonable amount to pay for a textbook. So I would get the 9th edition or newer. Maybe you can find a good deal on OfferUp or Craigslist or, any of these other online booksellers. Um, like I said, I don't think it matters if you're taking 181 or 182 or both of them. I think that your only option now is to get the whole textbook, and it's quite large. But if you take 182, you already have the second half of the book, so you don't have to buy another book. You don't need the Mastering Biology CD. We're going to use Canvas instead because it's free. Um, if you so a lot of students don't really read the fine print when they sign up for classes, but this class doesn't require it, but it's strongly recommended that you have at least 1 year of high school chemistry or 1 semester of college chemistry because it's chemistry intensive. Now, so students freak out when I say this, but you can still do well in this class even though you haven't had chemistry. It's kind of like if I had two students, or if I had a bunch of students and I gave them some sheet music to play Chopin or Mozart's Piano Concerto No. 7 or whatever, and I said, well, you're going to have a recital in a month. So one student has already had piano lessons for a year, and one student hasn't. Is it possible for the other student to learn this and do well in a month when they have to do their recital? And the answer is yes. But how are they going to get there? The way that they're going to get there is they're going to have to do a lot more practicing than the person that's already had chemistry, that already knows how to play the piano. So, it's totally possible. It's just that you're going to have to study more. This is a study aid. So this book is completely optional, but it has a lot of practice problems in it, and which is how you're going to get good at this. And I think it's like $30. So, I recommend it, but if you probably find the same things on the internet. It's just all located in one convenient source. The lab manual. So there's no lab manual this semester because we're completely online. Like I showed you, all the labs are on Canvas. So you don't need a lab manual. You will need high-speed internet because there's a lot of video and stuff like that. You'll need at least Windows XP, Java, different video viewers and things like that. Most people have this in their computer these days. The most important thing is that you'll need a computer and a webcam and a microphone. All right. So, like I said, that covers things that are small, molecular, cellular, organismic levels of biology. Small single cell things. We're looking at cells. Again, 1 year of high school chemistry or 1 semester college chemistry is recommended. You probably didn't see that, but it is. We talked about the grading system. So 4 exams, 600 points, 10 quizzes, 100 points, and then 12 labs, that's 300 points. So 1,000 points. And then it's a typical, so 900 to 1,000 is an A, 800 to 899 is a B, and so on and so forth. I don't give up makeup exams. It's too hard to do that, and especially in this situation, and to make an entire new exam for one student is unreasonable. So if you miss an exam, if you have a valid excuse, then I will allow your final exam to count twice. That way, the final will be worth 300 points instead of 150. Now, I caution you to use this sparingly because the final exam covers the hardest material of the whole semester. So it's not the best policy to blow off the first test and then try to do really good at the final. That generally doesn't work out that well. And everything in this class builds on everything else, and you'll see that. So to pass this class, you need a final and at least 2 lectures. The quizzes are under the quiz tab, and we already went over that. The lectures online and self-paced. I'll be paying attention to make sure that you're logging on and stuff like that. So, just try to keep up. The labs are all online, submitted to Canvas. Like I said, you have to do 12 out of the 14 labs. If you've done less than that, then I will drop you from the class because I feel like you're not participating, and these labs are important. And the lab material will be included on lecture exams. So some lab material will be on lectures. No makeup labs. So if you miss it, you just miss it and you lose the points. Like I said, you can miss 2 with no penalty. After that, I'll drop you. I accept 1 late lab, right? So you can tell me, whatever excuse you want, whatever happened, and then you lose 10% per day for the late lab. After 10 days, it's 100%. So you might as well not turn it in. You know how to get to Canvas. It's probably not a big deal. MCC is different than ASU and most 4-year schools where I can give you a withdrawal all the way to the very end. So if you're not doing well in the class and most of you will have to retake it anyway. The way I look at it, you already paid for it, and I can give you a W at the very end even after you get your final grade. I usually give the students a day to mull it over and decide if they want to withdraw or not. So I wouldn't be too quick to take a W. I would try to, go through the whole class, because even if you don't get a grade and you end up taking a W, you're still going to cover the material again, and practice makes perfect. So if you're going to do it twice, you're going to probably do a lot better if you do the whole thing twice than if you did half of it. if you just disappear, then I can't give you a W. So I'll give you a withdraw failing, which is a Y, and that will affect your uh, overall ability to retake the classes and things like that. So let me know what's going on. Don't just disappear and stop coming to class. Don't cheat. Um, I don't want to go to the dean's office. It makes me nervous, and I'm certain that you don't want to go there either over academic dishonesty. So don't do it. I've seen everything. It's like my kids trying to get away with stuff. If you have a disability, you can register at disability resources and you can get special accommodations. It has to be documented. I have to get an email from them. The class rules and visitation. I mean, I don't know what's going to happen. So I just left this in, but we're not going to be on campus, so you probably don't have to worry about this. Early alert program, so you can register this is completely voluntary. There's a veteran services provided by MCC. You can notify them or me of any special drop codes or anything you might have. And then again, there's this tutoring help that I showed you guys. So this is the in-person class schedule. This is the schedule that I cover in an in-person class. And so I just put the dates in here. Again, this is on your own pace. And the danger of this is that students tend to wait till the very last minute and then try to cram everything in. So it's back to the example of learning to play the piano. You have a month. If you wait till the night before to try to learn to play the piano piece, you're probably not going to do very good. And then you're going to get super nervous and you'll get test anxiety, which we would call piano anxiety, I guess. And so just make sure that you're prepared, right? If we're on if it's August 31st and you haven't started, you're already a chapter behind, right? Um, and if we get to Labor Day and you aren't finished with chapter 1 and 2, and that's all the stuff, right? PowerPoints, lectures, crosswords, study guides, all that stuff, then you're already behind like 2 chapters. And we try to cover a chapter a week. So try to keep up. Here's the exams and the dates that they will be. And this is where you should be in lecture for each of those dates. So that's pretty much it. If you guys have any questions, then you can feel free to email me at obermiller@mesacc.edu, or you can visit me during office hours drop in. So I'm looking forward to it, and hopefully you guys will learn something, and we'll have a great semester, and everyone will be safe. So I will see you guys on Monday during office hours if you want to come and ask me any questions about anything that we've covered or that you're not clear on. Otherwise, start watching the videos for the lecture on chapter 1, and then once you finish that, take notes just like you would in a regular class. Go over the PowerPoints just like you would in a regular class, and then start working on those crossword puzzles and those study guides, and make sure that you know all of the material for each one of those chapters. And then when you finish 1, go on to 2, and so on and so forth. And then by mid-December, we'll be done. So anyway, I'm looking forward to it. Again, if you guys have any questions, hit me up with an email or come visit me during office hours. And hopefully I'll talk to you soon. All right.